Glucose has a lot of different options when it first enters the body. It can get linked up into glycogen for storage. It can go the pentose phosphate pathway if you want NADPH and nucleotides. It can go the hexosamine pathway if you want amino sugars. Or what we commonly think about is it goes through glycolysis, breaks it down to pyruvate, generating ATP and NADH, which you can then cash in for energy in the electron transport chain. So glycolysis is really great if you want energy, but if you don't want energy, then you want to store the glucose. If you need NADPH, you want to go the pentose phosphate pathway. If you want amino sugars, you want to go the hexosamine pathway. But once you kind of go this fructose 6-phosphate to fructose 1,6-bisphosphate step, in glycolysis, that's basically committing you to glycolysis. The options are much more limited after this fructose 1,6-bisphosphate step because the other steps are kind of branching off either from glucose 6-phosphate or fructose 6-phosphate. And so we need to highly regulate this step where we convert fructose 6-phosphate into fructose 1,6-bisphosphate, which is catalyzed by phosphofructose 1 or PFK-1, so that we make sure that we only commit to glycolysis if we don't want to do those other things. And so if we have a lot of energy, we don't want to be committing to this step, but if we need energy, then we do. And so we want to regulate phosphofructose 1 highly. And we want to also regulate its counterpart, F16BP1, which is going to reverse the actions, go from fructose 1,6-bisphosphate, F16BP, back to fructose 6-phosphate, F6P. We want to regulate that as well so that we can promote gluconeogenesis, or the making of glucose, if we need to make glucose, or we can at least prevent breaking down glucose if we've got plenty of energy. So again, lots of fates before fructose 1,6-bisphosphate, fewer fates after fructose 1,6-bisphosphate. And so we want to really regulate this PFK-1 and F16BPase1 step. There are several key regulatory points in glucose metabolism. And this is basically one of the most regulated steps. And for those important reasons that we talked about, having fewer options afterwards, and the fact that this step is metabolically irreversible. So we actually need a different enzyme in order to go the opposite way in the pathway. We can't just regulate this based on the concentrations of these intermediates, so of F6P and F16BP. Those aren't really going to be the key regulatory factors. Instead, we're going to rely on allosteric regulators of the enzymes themselves, as well as covalent regulators of the production of the allosteric regulators. And so don't worry, it'll hopefully make more sense in a minute. So we're going to be looking at this step here. We're going to be looking at regulation of PFK-1 and F16BPase1. And we're going to be looking at the direct regulation by allosteric modulators, as well as the production of those allosteric regulators by this enzyme phosphofructose 2, PFK2, and its counterpart fructose 2,6-bisphosphatase 2, F26BPase2. You'll see that those are going to be regulated in part by hormones. So this is going to get a little gnarly, but bear with me, we'll get through it. Phosphofructose 1, PFK1, and fructose 1,6-bisphosphatase, or F16BPase, or you can also see it called FBPase1, they're reciprocally regulated. And so what that means is that the same signal is going to enhance the activity of one, while at the same time inhibiting the activity of the other. And so now you need to think about what sorts of signals you would want to tell you when to do what. And so think about when you want to do glycolysis and when you'd want to do gluconeogenesis. You want to do glycolysis if you need energy. And so you have a lot of fuel, but you don't have a lot of the usable energy from it. So you have a lot of glucose, but you don't have a lot of ATP. And so if you don't have a lot of ATP, well, what that means is you've got a lot of AMP and ADP because you've spent that ATP, you've hydrolyzed it, you've gotten rid of the phosphate group, and so now you have AMP or ADP. So AMP and ADP are going to be symbols that you have low energy, whereas ATP is going to be a signal that you have high energy. High energy, you want to inhibit glycolysis, and so you are going to inhibit this PFK-1. Low energy, you want to activate the PFK-1. And if you have a lot of citrate, that's another signal of high energy down the line, and that citrate is going to go and that is through like the citric acid cycle and stuff. If you've got a lot of that, you've got a lot of energy, you've got a lot of stuff building up further down the line. And so you want to inhibit the action to stop making more. And so those are going to inhibit the PFK-1. In the case of gluconeogenesis, however, you want to do that if you have plenty of energy. And so you want to activate this FBPase1 if you have a lot of energy, or you don't want it to happen if you don't have a lot of energy. So the same molecule we see citrate is going to be activating one, inhibiting the other, whereas AMP is going to be activating one and inhibiting the other. So you see this, the same molecule citrate is going to be activating this enzyme and inhibiting this enzyme, whereas the same molecule AMP is going to be activating this enzyme and inhibiting this enzyme. So reciprocal regulation. They're also reciprocally regulated by this molecule F26BP or fructose 2,6-bisphosphate. And now we get into the really gnarly stuff. First, let's talk about how the ADP and AMP and ATP are going to affect the activity of PFK-1. So what we're looking at here is the structure of PFK-1. And you can see that there are multiple binding sites for allosteric regulators. And so ATP, remember that was going to be a symbol of high energy, and that's going to inhibit this enzyme, and it's going to bind over here. And ADP and AMP, those are symbols of low energy status, and they bind at an alternative site. These are all going to be acting as allosteric regulators, so they're not binding in the active site, so where the substrate actually binds, but instead they're binding at other sites and affecting the activity that's happening in those active sites. When the ATP binds, this is going to cause a conformational change, so a change in the protein shape that inhibits the activity, but that's going to be relieved by binding of ADP or AMP to these alternative allosteric sites. And so what you're looking at here is on the x-axis, we have our concentration of our substrate, so our fructose 6-phosphate, and we have the activity of PFK-1. If we have a higher concentration of fructose 6-phosphate, we get a higher activity, regardless of whether or not we have ATP. And that's just normal like enzyme activity. However, what you can see is you get a lot higher activity if ATP levels are low compared to if ATP levels are high. So higher ATP, basically it's going to take more of the fructose 6-phosphate. We can say that it's increasing the apparent Km of the enzyme. It's going to take us more of the fructose 6-phosphate in order to get the same level of enzymatic activity, indicating that ATP is inhibiting this enzyme. Another key regulator is going to be this fructose 2,6-bisphosphate. And so fructose 2,6-bisphosphate, don't confuse it with fructose 1,6-bisphosphate. We make it from fructose 1,6-bisphosphate, but it's made in this alternative path. It's kind of the sidearm from glycolysis that's generating this regulatory product, fructose 2,6-bisphosphate, F26BP. And F26BP is going to regulate both PFK-1 and FBPase1. And so what you're going to see is that it's going to activate PFK-1 and it's going to inhibit F16BPase1. And so it's produced when you have a lot of fructose 6-phosphate, some of it's going to go down the sidearm, produce fructose 2,6-bisphosphate. The fructose 2,6-bisphosphate is then going to promote the activity of the PFK-1 and inhibit the activity of the F16BPase1. It's not quite like this, but you can think of it kind of like an overflow pathway. Like if you're building up a lot of this stuff up here, you want to promote the action down here. And so you take some of it and you use some of it to make this regulatory product that then can feed and tell this pathway, okay, speed up, speed up, speed up. We've got a lot of stuff building up above. So again, it's not quite like that. And instead, the activity of this PFK2 and this F26BPase2 are going to be highly regulated hormonally. And so these are going to respond to the presence of insulin, which is a signal of high blood sugar, and glucagon, which is a signal of low blood sugar, as well as signals of activity in the pentose phosphate pathway. So this is going to be highly regulated, the production of this, and then the produced product is going to regulate this step. So how does it regulate this? And so fructose 2,6-bisphosphate, again, so right now we're looking at the 2 position, and we're looking at the 6 position. And so 2,6-bisphosphate as opposed to just 1,6 bisphosphate, which would have phosphate at the 1 position instead of that 2 position. Here we have it at the 2, and the 6 position. And we call it bisphosphate, not diphosphate. Diphosphate would be if you had 2 phosphates in a row. Bisphosphate is if you have 2 phosphates, but on separate sites. Just a little technical naming note. So what this is going to do is it's going to bind to PFK-1 and increase its affinity for fructose 6-phosphate. So another one of those allosteric regulators. It's also going to bind to FBPase1 and reduce its affinity for its substrate. And remember, its substrate is going to be F26BP. So what we can see here is we have some more enzyme curves. Here, note that now we have fructose 6-phosphate on the x-axis over here. So again, we're looking at the PFK-1 activity. So this is kind of similar to what we were seeing before. But here we have fructose 1,6 bisphosphate because now we're looking at FBPase1 that's reversing the enzyme that reverses that step. Because remember, you need a separate enzyme because this step is metabolically irreversible. The delta G is too large and negative to easily reverse. So we need to use a separate pathway to get around it. Okay, so what we have here is we can see that in the absence of F26BP, it takes a lot of the F6P in order to get complete activity. But if we have F26BP present, well, now we're going to need less of the substrate in order to get the same amount of activity. And so we're decreasing the apparent Km, increasing the apparent affinity. In the case of our FBPase, well, now we see the opposite thing. We have stronger activity in the absence of F26BP and weaker activity in the presence of F26BP. And so this is another example of this reciprocal regulation where we have the same molecule activate one enzyme and inactivate or inhibits the enzyme that basically does the reverse reaction. So F26BP activates PFK-1, which increases glycolysis, and it inhibits FBPase1, thereby inhibiting glycolysis and promoting gluconeogenesis. So F26BP binds to PFK-1, activates glycolysis, binds to FBPase1, inhibits FBPase1, inhibits gluconeogenesis, allowing glycolysis to continue. So how do we regulate the production of this F26BP? So if we look at our chart, we can see that it's regulated by insulin, glucagon, and the X5P. And so X5P, what this is xylulose 5-phosphate, which is a product of the pentose phosphate pathway. So you can think about if you kind of have a lot of the pentose phosphate activity, maybe you want to shunt some of it back into glycolysis. And so if you have a lot of it, you're going to want to increase the production of F26BP. And you want to inhibit the inhibition of the PFK-1. So you want to inhibit getting rid of the F26BP and activate making the F26BP. You can also see it's regulated by insulin and glucagon. So remember, insulin says there's a lot of glucose. Let's let some in and let's break it down. Let's do glycolysis. And so if you've got a lot of insulin, you want to make this F26BP so you can increase the activity of this PFK-1 and promote glycolysis. Conversely, if you've got a lot of glucagon that says the glucose is gone, you don't have glucose to be breaking down. You want to be making glucose, gluconeogenesis. So you want to be inhibiting the production of F26BP so that you can inhibit the activity of PFK-1. At the same time, you want to increase the activity of F26BPase2 so that you can undo the making of F26BP, make fructose 6-phosphate, which then you can actually just use in glycolysis. And you want to inhibit the sidetracking of that fructose 6-phosphate into the production of the fructose 2,6 bisphosphate. So you want to inhibit phosphofructokinase 2. So how does this happen? So it's really complex in that it's kind of confusing in that these are both subunits of the same bifunctional enzyme. And so PFK2 and FBPase2 are both subunits of the same bifunctional enzyme. And that enzyme is going to be regulated covalently by phosphorylation. And the enzymes responsible, so the kinase that puts on the phosphate, CAMP-dependent protein kinase, or PKA, and the phosphatase that removes that phosphate group, phosphoprotein phosphatase, are themselves going to be hormonally controlled by glucagon and insulin. So we have hormonal control of the phosphorylation, which covalently controls the production of the allosteric regulator of the PFK-1 and FBPase1. And this regulator is made by the action of PFK2 and unmade by the action of FBPase2. This bifunctional enzyme is going to be active when it's dephosphorylated and inactive when it's phosphorylated. And so you want to phosphorylate it if you don't want glycolysis to happen, and you want to dephosphorylate it if you do want glycolysis to happen. Remember, when we have insulin, we've got a bunch of glucose, we want to break it down, we want glycolysis to happen. So we want to activate the phosphatase that removes the phosphate group, thereby activating PFK2 and inhibiting FBPase2, allowing us to make fructose 2,6-bisphosphate that can then go activate PFK1. In the case of our glucagon, well, here we're saying glucose is gone, so we're not wanting to break down the little glucose we have. Instead, we're going to just be wanting to make more glucose. And so we want to activate gluconeogenesis if we have that process going. And so what we want to do is we want to phosphorylate PFK2. That's going to inhibit PFK2, and it's going to activate the enzymatic subunit that is going to break it down, so the FBPase2. So phosphorylation activates the phosphatase portion of the enzyme, and it inactivates the kinase portion of the enzyme. Dephosphorylation activates the kinase portion and deactivates the phosphatase portion. And when I talk about the kinase and phosphatase portions, well, these are going to be of PFK2 and FBPase2, that bifunctional enzyme. And the phosphorylation and dephosphorylation of those is what's going to be controlled hormonally. So it's a bit gnarly, but it makes logical sense if you kind of sit with it for a minute. So the glucagon is going to act largely through that cyclic AMP-dependent protein kinase, PKA, and insulin is going to act largely through phosphoprotein phosphatases. If you look at glycogen metabolism, you see these same players show up again and again and again. In all of these cases, they're having the same sorts of effects, where the glucagon is going to be acting through PKA, promoting the making and release of glucose through the gluconeogenesis, glycogenolysis, inhibiting glycolysis. Insulin is going to be doing the opposite. It's going to be acting through phosphoprotein phosphatase. It's going to be promoting glucose uptake. It's going to be promoting glycolysis. It's going to be inhibiting gluconeogenesis. It's going to be promoting glycogenesis. It's going to be promoting those things that are going to take in blood sugar and break it down and use it for energy or store it. The final level of regulation of PFK1, and actually also of PFK2 and FBPase2, is going to be through transcriptional regulation. So changing the amount of the protein that we make by changing the amount of the messenger RNA that codes for that protein that we make. And so there is increased expression of these enzymes in response to insulin, and that's going to help regulate glycolysis. So lots of options when you first take that glucose in. So hexokinase, and so remember glucokinase in your liver, pancreas, intestines, that's the hexokinase 4 version is called glucokinase. Same thing, what it's going to do is it's going to phosphorylate the glucose to glucose 6-phosphate. That's going to trap it in the cell, prevent it from leaving. But now once you've got trapped it, you've got to decide what to do with it. You want to go the pentose phosphate pathway. You want to do that if you're going to want NADPH, if you want DNA, RNA, coenzymes. If you want to store the glucose, then you can go with glycogen metabolism. And both of those things could happen from this glucose 6-phosphate. If you take it then to fructose 6-phosphate, then what you can do is you can go and you can take it to the hexosamine pathway. You can make fructose 2,6-bisphosphate, or you can go down to glycolysis. Note too that these arrows are going to be reversible from the glucose 6-phosphate to fructose 6-phosphate. So you can kind of interconvert between any of these ones fairly easily. But once you commit to the fructose 6-phosphate to fructose 1,6-bisphosphate, well, now you have fewer options and you're basically committing yourself mostly to glycolysis. So we call this step the committed step of glycolysis, and we regulate it highly. We regulate the steps that actually does this conversion from fructose 6-phosphate to fructose 1,6-bisphosphate. We regulate the enzymes allosterically using signals of low and high energy. AMP and ADP are going to activate phosphofructokinase 1. And this is going to increase the activity of glycolytic activity. Conversely, AMP is going to inhibit the activity of fructose 1,6-bisphosphatase 1, and that's going to make it so that you're not doing gluconeogenesis. If you've got a lot of energy already, then you want to inhibit further glycolysis. You want to inhibit fructose-fructokinase 1. And so what you have is ATP, AMP, and ADP are binding to PFK-1 and affecting its activity allosterically. You also see that citrate is going to be another signal of the high energy status from further down the line. And if you got high energy, if you're building all this stuff downstream, you want to inhibit this PFK-1 step and activate the F16BP step. You also see that these are regulated by F26BP, and that F26BP is going to be produced in this sort of side pathway off of fructose 6-phosphate, produced by this bifunctional enzyme, PFK-2 and FBPase2. And that is going to itself be regulated hormonally through phosphorylation that is hormonally controlled by insulin, by glucagon, and by this X5P, so the cellulose 5-phosphate, which indirectly regulates the regulatory protein phosphatase that is then going to affect the phosphorylation of this PFK2 slash FBPase2 enzyme. And so it's going to promote the dephosphorylation, which has the similar effects of the insulin. So lots of different steps, but then you make this F26BP. And when you're making a lot of the F26BP, that's going to enhance the activity of PFK-1, inhibit the activity of F16BPase1, and thereby you have another level of allosteric regulation in this step. This is collecting information from hormones, from the pentose phosphate pathway, from the amount of activity up here, and it's going to therefore allow you to integrate a lot of different signals to control the commitment to glycolysis. Lots of fates beforehand, fewer fates after, and so that PFK-1 step is going to be the committed step of glycolysis.